Hematopoiesis is an essential biological process leading to the formation of red blood cells and immune cells. Both types of cell come from the same stem cells, the so-called hematopoietic stem cells, that emerge early during development from the walls of major blood vessels via the process called the endothelial to hematopoietic transition. Producing full potential hematopoietic stem cells in vitro remains a challenge, pointing to the relevance of reinforcing the fundamental knowledge of the process as it occurs in vivo. The zebrafish has proven to be an extremely valuable model for studying the developmental aspects of hematopoiesis. Indeed, zebrafish embryos are transparent, offering unique opportunities for in-depth live imaging. In this study, we reveal essential biomechanical features of the endothelial to hematopoietic transition using the zebrafish embryo imaged at unprecedented spatio-temporal resolution. In addition, in order to visualize the dynamic interplay between emerging stem cells and surrounding aortic neighbors, we developed an algorithm to unwrap the aorta into 2D cartography. We show that, as blood stem cells leave the aortic floor, they bend and contract along the antero-posterior axis with heterogeneous organization of contractile circumferential actomyosin, as well as of junctional complexes established with endothelial neighbors. This axis parallels the blood flow, raising the idea that the direction of the contraction, anisotropic since oriented preferentially along a specific axis, may be dictated by the mechanical constraints imposed by the aortic environment, and in particular, the blood flow. We thus addressed the influence of blood flow on the morphodynamics of the endothelial to hematopoietic transition by abrogating heart beating. Remarkably, the zebrafish embryo can survive for several days without blood circulation. Surprisingly, without any blood flow, the ventral side of the aorta still contains cells capable of performing an emergence. However, actin did not assemble properly, and intercellular junctional complexes became disorganized. The stem cells also emerged in the wrong direction, and some of them died a few hours after the release. Overall, these findings point to the influence of the mechanical forces exerted by blood flow in the formation of hematopoietic stem cells. Our work reinforces the idea that the mechanical constraints of the aortic environment should be taken into consideration when developing experimental settings aimed at producing hematopoietic stem cells in vitro.